Welcome to Rahbar Talk Show. Today, we want to discuss about the cancer. As you know, the prevalence of cancer and neoplastic disease have been increased during the last decades. Different etiologies and the condition can promote the occurrence of the cancer, in which the most of the people in the worldwide can be just afflicted by neoplastic changes. So be with us and see what happened. We want to discuss about the molecular pathway and different conditions that promote the occurrence of the cancer. Besides, we want to discuss about the conventional therapies and de novo modalities have been used to eradicate the cancer cell from our biological systems. So be with us and see what happened. Okay, Hassam, we want to just have a comments about the general terminology about the cancer and the condition that causes to the neoplastic condition. Can you help us to understand?
Actually, cancers represent a spectrum of highly adaptive evolutionary driven disease arising from the progressive breakdown of cellular regulation networks, such as uncontrollable dividing cell cycling and cellular signaling involved in their proliferation. But these cells are mutant or damaged from DNA section or segments that leads them to be a cancerous condition.
Okay, as I understood, you mentioned that the cancer cells can circumvent in different regulatory systems that control their cell cycle and their proliferation rate, and say they could not just respect all the, the limitation factor that control their dynamic growth, and even that these are some predisposing factor like a genetic mutation, and even that the metabolic switching from normal state to the abnormal state. That's the cause.
In general surface definition.
So the cancer cells at the first step there increase their numbers at the target tissue.
Yes. Actually, in the adverse and shapes, they are uncontrollable in their growth cycle, and they bind to the organs and maybe misregulate their function also.
Okay, great. Thanks a lot. Okay, Mr. Nazif Kerdar, how about the tumor types? Do you have any general items? How about the classification?
Yeah, as our friend mentioned, that these cells are going to be accumulate in our body and called tumors. So we have two kinds of tumors in our in this field. So one of them is solid and one of them is liquid. And liquid happens in our blood. So let's talk about the solid one. Solid have also two subtypes. Carcinomas and carcinomas and sarcomas. So sarcomas happens in connective tissues such as bones and muscles, and the last one happens in our lungs, breast, these kind of tissues and organs. So when it comes to the blood types of the tumors, liquid ones, it is leukemia and lymphomas.
Yes.
Leukemia happens in bone marrows and in some kind of immune systems tumors. They are going to produce lots of immune cells. This is kind of things. And also lymphomas happens in our lymph nodes.
In lymph nodes and as well the bone marrow. Both of them can be occur. Yeah, as you mentioned, great. We have the solid and non-solid tumors. In the solid tumors, these tumor cells have their own parenchyma. It's comprised of mostly the malignant and even benign cells, which are enclosed by the connective tissue, which is comprised of mostly of the type 1 collagen. So it's going to make some biological barriers to be to separate from the direct access of the immune cells. So even in terms of the blood and circulation, we have, as you told, we have two types of the tumors, leukemia and lymphoma. And so the leukemia are related to the certain lineage of the bone marrow progenitor. We call it myelogenous cells, which produce different types of the immune cells which are circulating in our body. And even in the context of the lymphoma, both the lymph nodes and even the bone marrow cells can be afflicted. Thanks a lot. Mrs. Mamegani, as you said, the most underlying mechanism which can orchestrate the occurrence of the neoplastic changes is the mutation in genetic levels. I mean that there's some changes like a drift and shift mutation in the DNA levels. How do you think about it? I mean, it's true.
Cancer is a genetic disease, but not necessarily in the sense of being always inherited, but because it's a change in genes which that control cell behavior, we call this genetic disease. And we categorize these genes in 3 classes, and we say them, and we can't say them or them like an accelerator or brake or mechanic for a car. The first classes is oncogenes. These are accelerator of a car. These genes are a positive regulator in cell, and they encode proteins which in normal cell controls cell growth and division, like growth factors receptors, intracellular inducers, and something like that. But a little mutation, like a mononucleotide mutation in just one allele in these genes, it's sufficient to make cancer in our body. For example, these mutations are gain of function mutations. For example, RAS is one of these proteins which is responsible in our cell division and cell growth. When we have a mononucleotide mutation in this protein genes, this RAS gonna be always this RAS protein is active all the time, and there is no need to any activator or anything, and our cell uncontrollably starts division. But there is 3 mechanisms here. First one is mononucleotide mutation. Second one is gene amplification. Here, gene copy.
So let me just elucidate and just scrutinize your previous comments about the single nucleotide mutation. You mentioned that the single nucleotide mutation can cause any alteration in structure of the protein. It makes some resistant to some degradation system maybe, and even that some of the allosteric changes and even there's some conformational alteration will be happening in the structure of that protein as opposed to just the trigger and accelerated molecular cascading. And so some of the metabolic reaction and even the biochemical reaction will be accelerated. So the cell exposed to just enhanced proliferation rate and just as we observe the increase of the tumor mass. And as you mentioned along with your statement as well, we have, absolutely, we have some different kinds of the RAS signaling proteins mutation like KFL4, which is very common in colorectal cancer patients. So which will be mighty great. And then
second mechanism I want to talk about it is gene amplification. In this mechanism, gene copies in our genome multiple times, and this makes overexpression of some kind of proteins like RAS, and that can make disease in our body. The third mechanism is gene moves around the location, another location, chromosomal location.
We have a displacement and even
replacement.
Replacement, somewhere.
We have 2 types of this. This gene can moves around under a strong promoter or fuses with another gene and makes oncogenic fusion protein.
Can you make any example about the mostly commonly, tumors in human with this mechanism, the latter one? We call it Philadelphia leukemia. It's a kind of leukemia, and it will be happen by this mechanism. Yeah, please.
The third classes of these genes are tumor suppressor genes. These are like a brake on the car. These genes encode some proteins which negative regulator of a cell. They make they repair DNA mistakes or make genome stable in our
correction,
cells.
yeah.
But if we have mutations in both of the alleles in these genes, they become inactivated and cancer happens in our cell and body. For example, P53, it's a genome guard protein. If both alleles become inactivated, this protein stop expression in our body. And if our DNA or gene have some mistakes has some mistakes and do not have some mismatches on it.
And you mentioned that it can suppress the activity of the oncogenes, and in some circumstances, even that these genes can be undergone some mutation, and so it will be deactivated. Yeah.
Yeah, and so our genes have some more problems and cause this cancer and a lot of disease. The third classes of genes are genome guard or DNA repair genes. These genes, like a mechanic for a car, they repair DNA mistakes, maintain genome stability, and something like that. And these genes, like tumor suppressor genes, when mutations happens in both alleles, and this mutation is loss of function mutations, they become inactivated. And when they become inactivated, we have a lot of mutations accumulate in other genes, especially in tumor suppressors and oncogenes. And this is cause disease.
Yeah, great. Very fruitful comments from your side. Great. And as you know, about over 90% of all cancer patients have alteration in metabolic status and even genomic profile. But we have also inherited cancer like a retinoblastoma. In other cases, we can just make an exemplify. And so these, how can I say, the cancer condition can be inherited to the offsprings. So very, very fruitful comments. Thanks a lot. Dear Mr. Ahmadzadeh, as you know, during the cancer's conditions, the tumor cells undergo some alteration in metabolic status, and even that in the genetic levels, in protein levels, and even some other metabolic profile. So we monitor any changes in these cells and compare it to the healthy cells to just see what happened in different signaling pathway or in different kinds of the molecule have involved in biochemical reaction. Can you make any brief descriptions about the hallmark and even that some biomarkers and even that the generation or the suppression of the specific factor which are related to the neoplastic conditions?
Yes, actually in 2000, there are two scientists named as Douglas Hanahan and Robert Weinberg, which these two scientists actually organized some hallmarks for cancer cells in order to distinguish them from healthy cells. And the first one is that the cancer cells can give out some signals in order to proliferate themselves independently from the outside signals, which is specific for cancer cells. The other cells, the normal cells, can't do this process by themselves. They need the activation from outside signals. But the second one, the second hallmark is about evading the growth factors or growth factor suppressors or even gene suppressors, as mentioned before, like P53, which I'm not going to elaborate more than this. These are genes that are like working as a brake for the cell cycle, and they are just for our cell arrest And by inhibiting these factors, cell cancers can make themselves proliferate more than this. The third one is about evading the apoptosis. Apoptosis is the process in which the cell dies. So in cancer cells, we have some signals that they give out of themselves. Sometimes it is like specific signals which are associated with these cancer only. So they don't die. The cell cycle is just going to multiply by itself more than this.
Yeah, and you mentioned that apoptosis is one of the background regulatory mechanisms to inhibit expansion of some neoplastic cells under a normal condition. This mechanism can just eradicate and eliminate some cells with undefined profile, like a cancer cell. So great. And even that over-acceleration of apoptosis is not good, because it can cause to the occurrence of the neurodegenerative disease or some other, malfunction cells. And so it should be just under control. By your comments, I just understood that the cancers can just be altered in a genetic levels, and some the suppressor and the cancer suppressor genes should be activated on the normal cells. But these genes are switched off, and by the occurrence of the neoplastic changes. And even that, the other supplementary mechanism like apoptosis or other cell death mechanism will be deactivated. So the risk of the cancer formation will be increased. Thanks a lot.
And the fourth hallmark is about the inability of replication in cancer cells, which is related to maintenance of the length of the telomeres. Telomerase is a protein structure at the end of the chromosomes, which are in every division of the cells. These protein structures are working like not damaging the DNA and helping the division. But after every division, these telomeres are shortened. But in cancer cells, we are having this process like maintaining their length, which is the real problem here. The fifth hallmark is about angiogenesis. Angiogenesis is a process in which the cancer cells give some specific signals to other parts of the body in order to make new blood vessels near them. Or also, I heard that in some new research, they have done about a research on breast cancer cells and saw that they can give some signals in order to distant neurons, in order to make them to come nearer to themselves and make their energy from that. I mean, the nutrients they need, they can take the nutrients for themselves by this process.
Great, very fertile comments.
Also, the fifth hallmark is about metastasis, which is the late stage of the cancer. In most cases, when this happens, the person has the lowest chance of survival because these tumors and cancer cells, by their biological molecules, as such exosomes, they are choosing a target site in order to go and spread to that. But it's still unclear that how these exosomes can choose that target tissue, and they are researching on it. Actually, in 2011, there are two supplementary added hallmarks to these cancer marks. One is Warburg effect, which is about the metabolism in cancer cells, which is altered, and they are using glycolysis in order to produce their energy needed. And the last one is the evading of immune system, which is one of the most important, and I believe from the research I have read from until now, that cancer is actually an immune inability disease, which is about actually our T cells. T cells are the in front line immune cells, which in every infection in our body, in every disease, they fight these infections. And in some cases, when they become much more than needed, they can even make autoimmune disease. So we have such another brake systems for these T cells called as Tregs. Tregs are T regulatory systems. They come and say that they make or inhibit T cells from activating much more than enough for our body. So our cancer cells are very brave because they can actually induce these Tregs and make them to activate and become more than enough in our body so that T cells become less than enough. When T cells become less than enough, our cancer cells, the cancer cells, are working more than enough. And
Great. Great. Actually, the cancer cells are a smart cell. They can just orchestrate different aspects of the biological reactions and biological phenomena. So these cells can just have a chance to just survive, and even that they can just expand and proliferate. In terms of the angiogenesis, you are right. Absolutely, the cancer cells can release different growth factors and pro-angiogenic factor like a VEGF, vascular endothelial growth factor, and even other factor. They can just dictate the active enhanced dynamic growth in endothelial cells, the mainly cells which are involved in the blood vessel formation. And in terms of the paracrine activity, as you mentioned, like an exosome and extracellular vesicle, you are exactly right. The cancer cells can just transfer different bioshuttles and different cargoes, different growth factor via these nanocyte particles and make some active cell-to-cell interconnections. So even that by sending these kinds of the factors, they can just dictate a distinct behavior on their surrounding cells, and even like a immune cell like T helper cells. They can just circumvent and even make some the shifting and even the circumvent their activity and make some mask on their external surface and just inhibit the invading and provoking to the side of tumors. You are right. Actually, very fruitful comments. Thanks a lot. Okay, Mrs. Bada, as you know, due to the massive changes in lifestyle, the human beings are exposed to different predisposing factors, components that increase the risk of the cancer condition. And in different societies, even in industrious countries as well in developing countries. In support of my notions, if you're going to compare the statistics with some previously released data, and even with the traditional cultures, societies, you can just find that the risk of the cancer will be have been increased by progressing the technology. How do you think about it? Is it true?
Absolutely, Professor. Actually, there are many factors which cause cancer. If we want to categorize them, the first one is the occupational and industrial materials, such as soot. Soot is the first agent which recognized as pathogen 7075. And actually, in radiology, which
Marx, highly important factors because the cancer are chemicals.
yes.
Okay.
such as microplastic, nanoplastic, which maybe we eating or breathing them every day, and we can control them. And I think it's very scary and worrying.
Yeah, great. In terms of the infectious particle, you are exactly right. In all items, absolutely, you are correct. And so the viral particle, and even virus, the active, they affect the genome and genomic balance, so make it some the site, how can I say, some of the active sites to their downstream the effectors. It can accelerate some of the biochemical reaction, and then you know what happens.
Yeah, also there are some special kind of chaperones called heat shock proteins, and especially 4 of them are really important, HSP90, 70, and HSP27, and also GRP80. And if the chaperones, if these lose their control, the gathered and aggregated proteins usually helps the cell to survive. Actually, this is the problem that happens. The factors, how to say, affect the chaperones affect the misfolded proteins, and these proteins change the expression of genes, and this is the start of the whole story of cancer.
Great, thank you. Thank you for your open comments. Mrs. Ojari, as you know, almost all types of the tissue in our body can be cancers. And so after occurrence of an initiation of the cancer conditions, different cells and different lineages can be provoked to the cancer parenchyma. So we exposed to different cell types which residing within the tumor mass. But in the clinical setting, we use just some drug targeting the cancer cell. And so these cells have inherently their own resistant mechanisms. And we have other cell types within the cancer parenchyma. So in the majority of the time and the majority of the cases, our therapeutic protocols are ineffective. So how do you think about it?
First of all, I'm thanking you for having me as a new member.
You're welcome.
As you told me, there are different reasons for different types of cancers, and different risk factors, different even 2 people with the same tumor has different profile in their genes and the environment of the tumor, and there are other problems too. For example, we have chemotherapy. When we use it, it can kill the tumor cells, maybe, if it's effective, but it can also, kill our normal cells as well. So, and we had a workshop with you, Professor, yesterday, and you talked about the solid tumors, and you said that it has different layers of cells in one solid tumor. For example, the center of it is necrotic cells. They die because of the low nutrition and the low oxygen, and but the cells around them, they used to know how to survive in that circumstance. So they are educated. They got educated. They get educating. So and if we use one method, for example, it may kill some part of that tumor, but some of them will be untouched because they know how to resist in that circumstance. So, and as I told you, we have another problem. Different patients have different profiles, and the microenvironment of the tumor is different. And the tumor can use immune cell for they can, for example, like Tregs or some macrophages, they can act for the help of tumor. So
instead of the
yes,
our body,
yeah. So in this situation, when there is so many risk factors, when there are so many risk factors, and there are so many reasons for this, disease, I think my interest is to use combined therapies. I think
yes, a good strategy.
For example, using chemotherapy plus, for example, cell therapy, like immune cells.
Immune-based cell therapies.
Yes, plus chemotherapy and I read some articles around that topic, and they were actually useful, I think.
That's all.
Yeah.
Yeah, by your comments, as I found that because of the different genetic makeup in each individual, so our therapeutic protocols are not could not just act similarly in all conditions. Even that there are some resistance mechanisms inside the tumor mass. I don't know, even either in levels of the tumor cells by their own selves, and even by controlling the conditions surrounding tissue and neighboring cells. So our protocols will not be effective in all cases.
I want to add something here.
Yeah.
You know, there are some methods like target therapy, chemotherapy, combined therapy, and the scientists, I think, they are learning to, and they are using a personalized, medicine. For example, they read about one patient and they want to use their unique, their specific therapy for that disease. But the problem is they cost too many. For example,
it's particularly for us.
Yes, for example, CAR T-cell therapies,
systems,
they are too expensive, or personalization therapies are too expensive. And I think one therapy is we can call it therapy when it's for all people in the world, for in different economical situations.
Yeah, there are also different risk factors that just limit and restrict the efficiency of our therapeutic protocols. Yeah, great. And so in personalized medicine, any changes and every distinct profile and traits of each individual should be defined at the transcriptomic levels and even proteomics. And so we could just decide which protocols can affect properly in that individuals. Yeah, great. Thanks a lot. Okay, as you know, the cancer is leading cause of death worldwide. So any debates and any discussion in this regard will be welcome. So because of its importance in a clinical setting and even in societies, we want to just continue our discussion to the different session. If you like this discussion section contents, you can just like, share, and even write down your comments. If you have any suggestion, please provide in our YouTube channels. Thanks a lot.